Hi everyone, welcome back to the channel. This is Dr. Asi, and today's topic is BSC 1010 General Bio 1 and Chapter 1 and Chapter 2. So let's get started. First, our textbook is Campbell Biology 12th edition. So if you haven't started, I would recommend actually getting the e-textbook, going to Canvas and clicking on course materials, and then registering with the Pearson, and start reading the chapters 1 and 2 in e-textbook. So also there are homeworks related to these chapters 1 and 2. So you can also work on these homework assignments, as well as you can also take the pre-test today and submit actually using Lockdown browser. So chapter 1 and 2, both slides are available for you in module 1, and also there is a syllabus in module 0. So if you edit the class later last week, you can still have all these materials and start reading. You don't need to wait until next class, so you can start actually reading and working on the materials today. So let's get started. Okay, we already covered this chapter 1 and already last week. So chapter 1 is about themes of biology and scientific inquiry. So we have these levels of biology, starting with atomic level to molecular level, organelle level, cell level, tissue level, individual, population, community level, all the way to ecosystems and biosphere level. So biosphere in this picture is the largest level, which is all life on Earth plus environment they inhabit. They actually call biosphere. Ecosystem is one level down, which is all living organisms in one area and non-living organisms. That means there are several ecosystems. Communities and all organisms in a particular ecosystem. Population, all individual of same single species. Organisms, individual living entity, and it goes on like that. So we have chapters on each of these items. But there is one of these themes that actually is not a chapter, but it's a concept that it's number 1 concept here, which is called emerging properties. If you were in a class, you might remember from last week, emerging property results from a arrangement of interaction between parts. And so the whole new things come out as an emerging property is greater than its parts. So something new come out. So when you're studying that, pay attention to that. And an example was, if you take sodium and chlorine and if you add them together, you make something, sodium chloride, like table salt, which is actually has more properties than just sodium and chlorine individually. So, and emerging properties come out, same as water. So keep studying on chapter 1. Chapter 1, 11 themes of biology. You can see here cell, DNA, structure, function, regulation, interaction with environment, evolution, very big one, systems biology, inductive reasoning, and genomics. These are 11 themes in biology that actually use commonly in biology. So there is more information here about emerging properties. You can read about cell structure and function. Cell is the smallest unit that actually can function by itself, so independently. That's why cell is very important, and we have whole chapter 6 on just the cell itself. And cells, we divide them in 2 parts, 2 groups of cells. Prokaryotic, which is the small cell you see here, and eukaryotic, which is these large cells like you see here. But we have a chapter on this specifically, but this chapter 1 has a little information like what do they have in common, these 2 cells. Well, they all have plasma membrane, they all have cell division, they all have cytoplasm, and they all have ribosomes, and they all have chromosomes. You need to remember these things they have in common between eukaryotic cell and prokaryotic cell. Okay, so and let's continue. We have more information on cell in upcoming chapter 6, which is not yet. So we have a number of chapters before that. Okay. Now let's look at some other topics in chapter 1, which is of course chromosomes and DNA and genes, very, very important, and we have several chapters on these. DNA molecule, as you probably already know, like any other macromolecule, is made of smaller subunits, like in this case AGCT. These are called nucleotides, and they are in this double helix. And if we actually decode these AGCT sequence, that's called DNA sequencing, and we can actually print that out and look at the sequence and see some patterns and specific genes in the DNA. Okay, so genome is more like a big picture of DNA of whole organism, and look at, like, see patterns in a whole organism level. DNA information flow goes from DNA to mRNA, from mRNA to protein, right? So we will see these in coming chapters, and very, very important information flow. And of course, there is feedback. We will see negative feedback versus positive feedback. Negative feedback is if the end product goes and slows down or stops or slows down the whole reaction pathway. This is called negative feedback. We've seen 11 themes, but also in order to classify organismal level, we use from species to genus, family, order, class, phylum, kingdom, and domain. Domain are the largest units. Phylum, very, very important. Kingdoms like Plantae, Animalia, fungi, and this is more covered in Bio 2, not so much in Bio 1. Three domains: bacteria, eukarya, archaea, and eukarya are the three big domains. And of course, eukarya has kingdoms like Plantae, fungi, Animalia, and Protista. So evolution is another very important topic that actually unites, like, one of the 11 themes of biology, and but this is also more in Bio 2, as well as genetic trees, like phylogenetic trees, like you see here in this picture. Another topic in this chapter 1 is a scientific method, which is a problem-solving technique in biology. So it has this order: you come up with a question, you form an hypothesis, which is an educated guess, kind of you try to find an answer before you actually start doing an experiment, tentative answer to your question. Okay. So for example, if your question is, okay, why the flashlight doesn't work, and what is the reason flashlight doesn't work, right? Your hypothesis might be the batteries are dead. This might be your hypothesis. And you might go and test that and find enough data and see if you can reject this hypothesis. If you cannot reject this hypothesis, then you accept this hypothesis, and you basically come up with a conclusion that this is the reason actually causing this flashlight not working. But there are other similar topics besides hypothesis. So a theory, for example, in biology, is a stronger explanation based on extensive evidence and many hypotheses supported this theory. And in chemistry, you might see law or principle, which is a description of observable phenomena. Okay, so that's chapter 1. And you can actually read the whole chapter in your book, and you can actually work on your chapter 1 homework. Now, I'm going to move to the chapter 2, which is basically inorganic chemistry. So chapter 2, which is titled Chemical Context of Life. So we have two big goals here: understanding basic chemical principles affect living things like biology, and learn about structure and function of atoms, elements, compounds, and molecules. So we have vocabulary here. If you are not so strong in background, if you don't have a strong background in inorganic chemistry, um, maybe you might be taking inorganic chemistry right now or general chem 1. It might be very, very useful. So key concepts here are matter, which consists of chemical elements in pure form and combinations called compounds. Elements' properties depend on the structure of its atoms. The formation and function of molecules depend on chemical bonding. And then chemical reactions make and break chemical bonds. So here is a little summary of chapter 2. Matter is defined as made of chemical elements, pure or combination, which is called compound. Again, I just mentioned chemical bonds between the atoms actually make things happen. They can break or make in chemical reactions. So, of course, general inorganic chemistry, chapter 2, starts with periodic table, which with 118 elements. So you all know periodic table. It's designed by a scientist actually who likes card games, and designing is like in card games, like suits. Each columns is a suit. It's sorted by weights of the elements. So, and today we still use this system, which is called groups: group 1, group 2, group 3, group 4, group 5, group 6, group 7. And then there is group 0 at the very right end, which are called noble gases. Noble gases are inert elements, and they have closed shells. So they basically they don't do anything with anybody. So okay. Now same number of valence electrons. So we are gonna see each group, they are in that specific group, like group 1A, because those elements actually in each group have the same number of electrons in their outer shell. Okay, the group 1A, everyone has 1 electron. That's so that's why they are in group 1A. Group 2A, they have 2 electrons in outer shell, and then it goes on like that. Okay, group 3A, 3 electrons, 4A, 4 electrons, 5A, 5 electrons, 6A, 6 electrons, 7A, 7 electrons in their valence shells. And remember, its goal is to reach the 8 electrons to complete. So don't forget that. So even though we have 118 plus elements in the periodic table, there are 25 of them that are important in biology. Okay, so, and 4 of them, carbon, hydrogen, oxygen, nitrogen, they make the 96% of the living matter. So without 4% phosphorus, potassium, calcium, sulfur, they are also highlighted in purple colors here. So these are called essential nutrients, as well as some beneficial ones, again, depending on the living organisms. So we divided these 25 into groups. One group is called macro, M-A-C-R-O elements, which are these large ones, like nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, carbon, hydrogen, oxygen. But also there is another group called trace elements. They are smaller amounts required, which are like iron, zinc, manganese, chlorine, iodine, nickel, and goes on, and molybdenum. So there are two examples here in this slide, which is iron, of course, very, very important, required by all organisms. Iodine, another one, which is everyone needs to take 0.15 milligram iodine daily. If you don't take that, you can see it might cause iodine deficiency and goiter. You can see in this second part of this figure here. So this can be actually avoided if you're using actually salt here in the US that that they have iodine in it. Okay, so um, so if you look at the atoms, so each of these elements made of the atoms or atom of that specific element. So and inside the atom we have three parts: neutron, protons, and electrons. Neutrons have no charge, protons positively charged, electrons negatively charged. So we have two terms. One is so each element in periodic table has two numbers, one large number, one small number, right? Like carbon. Carbon has 6 at the top and 12 at the bottom. 6 is called atomic number, and 12 is called atomic weight or mass. Okay, so atomic number is same as proton number, um, which is fixed number. It always fixed, but atomic weight is proton plus neutron, which is an approximate atomic weight number, which is the large number in each element. So helium you see in this example, 4 is their mass number. Estimated atomic weight is 4 daltons. Okay, the carbon among 118 elements is the king of periodic table and can form 4 covalent bonds. Why 4? Because carbon is in group 4. And group 4, we know, has 4 electrons shell in their outer shell. That means they can form 4 bonds, 4 covalent bonds, carbon. Okay? That's nothing to do with its atomic number or mass number. It's more about how many electrons is in outer shell. How do we know that? Its group number tells us how many electrons are in its outer shell. Carbon is 4, nitrogen is 5, oxygen is 6. Okay. So why atomic weight is estimated but is not an exact number? Because in the nature there are more than one form of these elements. So carbon, there are more than one carbon available. But in periodic table, there is only one carbon representative. So they are called isotopes of each element. So two atoms, same element, different number of neutrons. So carbon, for example, there is carbon 12, which is a most common one, 6 neutrons, carbon 13, 7 neutrons, carbon 14, very rare one, 8 neutrons. Okay, so these are all three like different isotopes of carbon. So a lot of them are stable isotopes, but there is one group of isotopes called radioisotopes. They decay and then they actually emit very high energy. Carbon 14 is can be considered one of those. They can be used in biology, an effective way to visualize things like you see imaging in biology or dating fossils of thousands of years old fossil records to find how old that is and how long ago that animal was actually passed, and that can actually be very, very useful. Why they are used for that purposes? Because these radioisotopes have very high energy. Okay, so the electrons on the other side are in the shell. So they are also called energy levels, electron shells. The farther away you go from the energy shell, the more potential energy they have. Okay, that's why they can stay farther away. Electrons can only change their position. They can switch to the lower level by absorbing or releasing energy, vice versa. Okay, so they have potential energy. Chemical behavior of atoms depending on the electron shell. Electron shell determines they are actually determines which row they are gonna be. One shell is the first row, two shells, second row, three shells, third row. You can see four shells, fourth row. Okay, so these are energy levels. Moving from left to right, each element adds one extra electron. Like you see, this one has one electron in the outer shell, but this one has 7 electrons in the outer shell. Okay, this is also called valence shell, and we focus on valence shell most, okay, in biology. So the bonds, okay, atomic bond, atoms, they can actually can be combined using this chemical bonding to form molecules. These are called chemical bonds. They can be strong, like covalent, ionic, or weak, like hydrogen, van der Waals, and ionic. Okay, ionic is in both groups because ionic in water is weak, but ionic in crystals is strong. Covalent bond, very important bonds that they based on sharing valence electrons. If two hydrogen get so close to each other, they can share these valence electrons and make H2. If we write it like this, we can write it several different ways of this new molecule of H2. If we just write it like with typing, texting like H dash H, that's structural formula. If you're typing H2, that will be molecular formula. Or if you're drawing with a drawing program like this, we can do that too. These are all exactly same thing. Okay, so same thing here, methane, which is CH4. There is a term called electronegativity, an atom's ability to attract and hold their electrons. How strongly they can attract and hold their electrons. So there are some elements, they are very highly electronegative. Fluorine is 4, very strong. Oxygen 3.5, pretty strong. Nitrogen 3, strong. Carbon 2.5, medium strong. Hydrogen, it's only 2. That's actually not very strong. Okay, so if you put these in a table and groups, nonpolar covalent bonds, their electronegativity are low and the atoms share electrons equally. But polar covalent bonds, they have high electronegativity and they don't share electrons equally. Okay. A molecule consists of 2 or more than 2 atoms held together by covalent bonds. It could be a single bond sharing just 1 pair of valence electrons, or double bond, 2 pairs, triple bond, 3 pairs. It goes on like that. Okay, so there is this chart, but we are gonna see them in the coming up, like after these different bonds. Ionic bond is a bond that actually has basically is not sharing, but it's really stripping electrons from the partners, not sharing but taking it away or giving away. The best example of ionic bond is like you see from your book, sodium chloride. Sodium is a group 1A element, which has 1 extra electron. It's willing to give it away. Chlorine is group 7A. It has 7 electrons. It just needs 1 more and willing to take 1. If they get close to each other, they do that. Now sodium give away 1. It becomes positively charged because it's always good to give. Chlorine takes, which is not very good, and it's negatively charged. Chlorine in this case called anion, and sodium called cation. And what they form now is an ionic bond. Not very strong, but it's a bond. It's called ionic bond. There is also hydrogen bond, not too strong and too common. It's basically when a hydrogen atom covalently bond to a one electron negative atom of another molecule. Like you see here, water, its hydrogens on the sides get close to this NH3 ammonia and connect itself to the nitrogen here. Very strong electronegative element. This is called hydrogen bond. There is another bond called van der Waals. It's not a really bond, but it's called interactions. Uh, that's basically not a covalent or ionic, but it's some other type of interaction. An example here is this gecko. Its toes, you can see geckos actually climbing on the walls, not because they have some superpower. Their toes have 500,000 hairs, and then that can actually bond it with molecules of wall surface and can hang on wall using just one toe. This is van der Waals. Another topic here is uh, molecular shape. Molecular shape determines how biological molecules can be recognized and respond to one another. So sometimes molecular again shape determines function, but it can take advantage of this molecular shape because a shape is also goes and connects itself to a receptor in the cell. So here is an example here. There is a natural endorphin or morphine which their shape look like each other, and they can go and use the same bind to the endorphin receptors. Insulin, a very important hormone in human animal system, which actually makes the glucose uptake in the liver and other muscle cells. Basically, if that's because of its shape, we can actually give external insulin, and then it can go and connects itself to that insulin receptors and open up the muscle cells and move the glucose to the muscle cells. A synthetic insulin is a possibility which a lot of people takes with type 2 diabetes or type 1 diabetes. And lastly, chemical reactions make and break these bonds. So how they have reactants at the beginning, and then the reaction takes place, and then they produce products until they reach chemical equilibrium. So in this chapter, basically, there is just introductory general bio 1. General chemistry 1, which you might be taking. Again, atoms, proton, neutron, electrons, atomic numbers, mass numbers, electron shells, isotopes, valence shells, polar and non-polar molecules. Then charges, protons are positively charged and electrons are negatively charged. The proton and neutrons are 1 Dalton in terms of their mass and their location, the nucleus, but electrons, their mass is negligible and their location is orbitals in an electron shell. So we have an e-textbook again, can be accessed through your Canvas course materials, and you can register there. Please do that soon without delaying that. And start working on your chapter 1 and chapter 2 homework and take the pretest today if you haven't yet. So we will continue chapter 2 in the next class. Just to recap, again, this was chapter 1 and 2 from Campbell Biology 12th edition. And this is Dr. Hasse. Thanks for watching. Please feel free, comment, like, and subscribe. See you in next video and happy studying.